Good morning, everybody, and thank you for the introduction and the kind words that you've had for me. So today, first of all, I would like to thank the organizers for giving me the opportunity to come here and talk about my research to you. But I have kept this talk more generalized because I had an impression that most of the audience here are students, and I would like to take this opportunity to make them aware about an extremely important problem that the world is now facing, and that is antibiotic resistance. So the topic of my presentation is antibiotic resistance, the past, present, and future. So you will have glimpses of the past, and then we will also look at the situation which may happen in future. But primarily my talk will focus on the present day scenario of antibiotic resistance and how we need to tackle it. And as I do that, I will also be giving you an idea of the kind of work that we do at JISU on antibiotic resistance. Okay, so if you look at these two graphs here, both of these graphs look very similar and actually represent two burning problems that the world is now facing. One is the problem of climate change, where you can see that across the years, there is this climbing, rising temperatures across the world, which we know is a huge problem. And the other, of course, is the problem of antibiotic resistance. Again, this graph shows the trends in the antibiotic consumption for the last, from 2000 to 2010. So both of these are burning problems, but the way we are dealing with it seems to be extremely different. So this is the COP26 meeting. We know that the climate change is getting a lot of attention, with global leaders from all across the world coming across and discussing the problem of climate change. But in contrast, antibiotic resistance is still, the awareness about the problem still remains only with the clinicians or the scientists, and there is much less awareness about this problem across the general public. So we'll see how, as I said, my topic will move across the past and then future and then come back to the present. So we'll be looking at this problem from various aspects. The first is the years around 1928 to 1960 and also 1970. This was considered to be the glorious years of antibiotic research, primarily because this was a time when most of the antibiotics that we have in clinical use now were discovered. So this, the landmark discovery of and the development of penicillin took place between 1928 to 1945, where in 1928, Alexander Fleming discovered this antibiotic, but it took years to bring this into clinical use. And we all know that this particular discovery has been the impact it has had on saving lives has been huge. So this was the start of a period when we could see that infectious disease, there was some method of really fighting back infectious disease. Now, once we had, as I said, these years were really the glorious years, and after the discovery of penicillin, we had a number of other antibiotics which were coming into the forefront, and that was due to one particular person, Selman Waxman, who actually identified what we now call the Waxman platform. And he, as is written here, the golden age of antibiotic discovery began with the Waxman platform, where he systematically tested soil microbes for identifying newer and newer antibiotics. And this particular drug, which was considered to be the miracle drug, and truly because it saved so many lives after the identification of the different antibiotics, we could fight back a number of infectious diseases. So, as I said, we had a number of antibiotics during this period, and quickly we had quite a number here, but slowly as we were getting more and more antibiotics for the treatment purposes, it was thought that probably we have won the war against infectious disease. So once that happens, slowly the discovery process began to lag behind. But even then, we had quite a number of new antibiotics which came into practice and which was used for patient care. But after that, the discovery, the phase of, there was this discovery void which started happening after 2000. And we now do not have too many antibiotics, newer antibiotics, and there are several reasons for that, which we will hear about. Now, once antibiotics were used, slowly, again, this particular picture will give you an idea that once antibiotics were used, the resistance to these antibiotics were also getting noticed. And Alexander Fleming himself mentioned that once you have an antibiotic, you can soon expect that there will be resistance to this particular antibiotic, because we know that microbes can very easily modulate and change themselves to adapt to situations which are harsh for them. And that came in, and slowly, as you see here, you have a particular antibiotic, and then within a year or so, you have resistance to these antibiotics. Now, let's move forward. Let's do a fast forward to 2015. And this is an excellent paper by O'Neill, who was given the task to actually predict how big the problem of antibiotic resistance was. And he came up with his report in 2016. And this is one of the most important messages from his paper that we have here. And he showed that in 2050, there will be about 10 million deaths which are attributable to antimicrobial resistance. So this was his prediction. And the number of deaths will be even more than what you see due to cancer or many other lifestyle diseases like diabetes or other infectious diseases like cholera or measles. So this is a scenario which we just cannot make it happen. So let's see what the situation is now. So we come back to the present day, and we will be discussing now in details about the situation about antibiotic resistance. So this was a report, a statement from the World Economic Forum that concluded that arguably the greatest risk to human health comes in the form of antibiotic-resistant bacteria. We live in a bacterial world where we will never be able to stay ahead of the mutation curve. A test of our resilience is how far behind the curve we allow ourselves to fall. And if we really allow ourselves to fall behind the curve, we know what the situation can be. Because these are drugs which have saved lives, which have like a dose of 5 days can cure you. But if these drugs stop working, then you will be losing people to very simple diseases like even a cut, a wound in your hand. So that is something which we cannot allow to happen. Okay, now coming to some of the antibiotic resistant bacteria. Now, some of the bacteria, if you will see when you look into the literature, have had the ability to mutate quickly, and they have also had the ability to acquire antibiotic resistance genes very quickly. And these are a group of bacteria that we call the Enterobacteriaceae, which includes Klebsiella pneumoniae, which includes Escherichia coli. Then we have Acinetobacter baumannii, we have Pseudomonas aeruginosa, we have Enterococcus, we have Staph, and of course we have MT. So these are some of the bacteria which play a very critical role in the spread of antibiotic resistance, and they also play a very significant role. And because of their importance, the World Health Organization has now brought in a priority pathogen list so that the world knows which are the critical pathogens. So these are the ones which are the most problematic ones, and then you have the high priority pathogens and the medium priority pathogens. And if you remember in my last slide, I showed the critical pathogens were carbapenem resistant Acinetobacter. So Acinetobacter, which are resistant to the antibiotic carbapenem, Pseudomonas similarly, and Enterobacteriaceae, which are carbapenem resistant and also resistant to the third generation Cephalosporins. Now, this particular figure, again, an excellent study which was published in 2022 in Lancet, was to estimate the burden of antibiotic resistance. So O'Neill had given us a prediction of what would be the burden in future, but this is a present day estimate of the global deaths which are attributable to bacterial antimicrobial resistance, and they have given a combination of the different bacteria and the drugs and the number of deaths that are happening due to it. So anywhere you see the color moving towards red, that is the orange or the red colors, are counts which are either greater than 100,000 or in the range of 75 to 100,000. You can see that some of the bacteria, Acinetobacter, and then you have Escherichia coli, you have Klebsiella pneumoniae. Again, you remember the WHO priority list. All of these were in the WHO priority list. The same paper also had these heat maps, which showed the rates of resistance for particular pathogens. So this was a picture for the Staphylococcus aureus, methicillin-resistant Staphylococcus aureus, and you can see again, as you move towards orange or a red color, you know that these are countries. India is one. You can see the rate of resistance to this particular drug in India is around more than 50%. Now you see India is in a color red, and this is a this is a heat map for the fluoroquinolone resistant Escherichia coli. And you can see that resistance to fluoroquinolones is extremely high in India. Take any organism, this is a drug which seems to have around 80 and in some cases around 100% resistance. Now, so this was the global scenario. If we look into the scenario in India, because India has a huge population and India also has a very high drug resistance index. So this was a calculation which was done by the Center for Disease Dynamics and Economic Policy, where they carried out an estimate of the drug resistance in different countries. And India has a drug resistance index of 71, which is the highest across the world. So antibiotic resistance is an extremely big problem in India. Again, we come to some graphs, one of which you've already seen, rising curves, which show that the consumption of antibiotics in India is extremely high and keeps on growing and growing. And the rates of resistance to some of the drugs are nearly 90% or so. And most of the drugs will not work in case you are using them for the patients. Now, this is a very simple diagram, and probably if you are a little bit into microbiology, you will know how antibiotic resistance develops. But quickly, if we just go through it, because I will now move on I have already given you a scenario of the status of antibiotic resistance across the globe and in India. And slowly I will now move on to the work that we do on antibiotic resistance. Now, whenever you are using antimicrobial products to kill bacteria, some of these bacteria, always they are the clever ones, so they will change themselves in certain ways due to which they can resist the antibiotic. So most of the bacteria will die, but one or two of them will still be alive. And these are the ones which will quickly multiply, because we know bacteria has a very short span of multiplication. So they will multiply, and these are the resistant microbes, which again will have certain genes which they can pass on to their progeny, or they can pass on to other species, and hence antimicrobial resistance keeps on increasing. A little bit on the different mechanisms of resistance. Again, something most of us here may be knowing is that the problem is so huge because there are several mechanisms of resistance to each of the different antibiotics that we have, and most of these are, there are several commonalities, but there are also several differences. So you can have bacteria which does not allow the antibiotic to move into the bacterial cells. So they are resistant to antibiotics because these bacteria have lost certain proteins which are called porins, which allow the entry of the bacteria. You have across different species, different efflux pumps, which have the capability of effluxing the antibiotic which enters the cell. So that is again another mode of resistance. You also have antibiotic modifications. You have target modifications so that the antibiotic cannot come and sit on the target site, and hence there is resistance. But one of the most important resistance mechanisms, why I say important is because this is a mechanism which can quickly spread from one bacteria to the other, and that is the presence of certain antibiotic activating enzymes, which can go and inactivate the antibiotic. These genes are primarily born on plasmids, and these plasmids can move on from one bacteria to the other very quickly, and hence you can have the spread of resistance is very quick. Now moving on to the work that we do. It is extremely difficult for me to describe in detail the work that we do. We've been carrying out work on antimicrobial resistance for nearly last 20 years now. But I will just give you glimpses of the work so that you can understand how the research is being carried out. So most of the work that we do in our laboratory is focused on neonatal infections, or what we call neonatal sepsis. So these young ones, after they are born, the first 1 month of life is called the neonatal period. And these neonates, because they are very vulnerable, some of them are of low birth weight, are premature, they are extremely susceptible to infections. And when they are infected with bacteria that are resistant to certain drugs, it is extremely difficult for us to treat these neonates. So that is the reason why we've been working on neonatal infections. There are quite a number of different modes of acquisition of this resistant bacteria, which can happen from the mother during the process of birth, which can happen from the mother during the period of nursing, from other caregivers. But one of the most important source of infection, of course, remains the hospitals, because these are the places where antibiotics are used in huge amounts, and antibiotic resistant bacteria also thrive there like anything. So this newborns who are of low birth weight, premature, need to stay in the hospital for a much longer time, and during which they may be exposed to drug-resistant bacteria. Again, an extremely important reason of why we work on neonatal infections. If you look into this graph here, you will see that the mortality rate across the globe for the under 5 mortality rate is declining at a very steady rate. The infant mortality rate is also increasing extremely fast. But in contrast, the neonatal mortality rate, the decline has been not very steep. So UNICEF has already declared that though there has been enormous progress in child survival, but greater focus on newborns urgently is needed. Now, this is a data from our lab where we look into the antibiotic susceptibility pattern in one particular organism, which is Klebsiella. And this is the data for nearly the last 15 years. And you can see that for most antibiotic, the rates of resistance are very high. And one particular antibiotic, which has seen a very steep rise for the last few years, has been the carbapenems, which includes ertapenem, meropenem, all of these drugs, which are considered to be the last resort drugs. So they are only used for patients who are seriously ill. But if you look into this graph here, you will see that resistance to such drugs, which are considered to be last resort, are also rising and keeps on rising with the years. This was looking into the different resistant genes which are responsible for this resistance to this drug, and we find particularly a gene called the New Delhi metallo-beta-lactamase, which is a gene which is present very frequently in Klebsiella, E. coli, Acinetobacter. So it's a gene which keeps on moving from one species to the other. This gene has been extremely high across the years. There are other genes which are also high, but this has kept on moving for several years now. So we, as I have already discussed in my slides, when we looked into the past, one particular event which all of this we've nearly talked about, but we haven't talked about the identification of this particular gene I just now mentioned, the New Delhi metallo-beta-lactamase, which was identified in 2010, 2009, during late 2009, and within 10 years, this particular gene has now spread across the globe. Now, this particular enzyme is a metallo-beta-lactamase, which can hydrolyze different beta-lactam antibiotics and can also hydrolyze drugs, which, as I discussed, are the last resort ones. So during the time when this particular was getting identified, we were also doing some work with rural hospital in Shuri, and this is a level 2 sick newborn care unit in the district hospital there. So we had collaborators, and they this is one event which I want to talk about, because this was something which remains one of the most important work that we have done, because it affected a number of lives at that point of time. So this particular unit was having a very high prevalence of drug resistance. And as a team, we wanted to conduct a point prevalence survey, where we go and we collect samples from different parts of the SNCU and also from the babies, and we wanted to survey what is the bacteria that is present. Within a few days of that survey, 4 neonates died due to septicemia. And I've just brought the whole story into one slide because it's difficult for me to describe everything in details. But after we carried the survey, within 10 days, we heard that few neonates had died in the particular unit due to septicemia, and these were all newborns. And from these newborns, from the blood of these newborns, we isolated Escherichia coli, and we also isolated and identified the gene NDM, which had just been reported in 2009. We were also doing the work at that point of time. We identified this gene in the blood isolates. Then what we did was we looked into the bacteria that we had collected during the time of survey, and we found that there were 2 different kinds of Escherichia coli, clonal type A and clonal type B. And this clonal type B was having the NDM gene, whereas the clonal type A did not have the NDM gene, was susceptible E. coli. And look at the E. coli which has actually infected the neonates. It was the clonal type A, but it had now acquired the NDM plasmid, probably from the clonal type B. And this was a situation which we faced, where we could show that how quickly a particular resistance gene can move from one bacteria to the other and then create a real havoc in a neonatal setup. We published this paper in a Journal of Antimicrobial Chemotherapy, and we talked about how this transmission took place, and we immediately talked to the clinicians, and the infection control practices were reinstated, and you will know how important that is for saving lives So infection control practices were reinstated quickly, and after that, this particular outbreak, which could have become extremely big, was died down within a few days. Now, as I said, the plasmids play a very important role. You know that these plasmids are what we call the extra chromosomal DNA. So you have chromosomal DNA, but you also have these plasmids, which in the clinical isolates, if you see, are not small plasmids. They are huge plasmids with large number of genes. And once they spread from one particular bacteria to the other, they can quickly transmit any gene. So as I said, the earlier study was with the New Delhi metallo-beta-lactamase, but this was a study which we have still not published, a work that we are carrying out, and this focuses on another gene, which is the MCR, which is known as the resistance gene for colistin. And again, we carried out a work. This was looking into the transmission of these genes from mothers to babies, because we know that the mother can become a huge source of spread of infection to the babies. And these were plasmids which we have, because of the advent of the whole genome sequencing facilities. These were large plasmids around 200 kb, 250 kb, which were sequenced, and see how homologous these plasmids are, how similar these plasmids are, which were identified from several different mothers, and they look so similar. So again, an evidence of how fast the resistance can spread. This is from another study of hypervirulent Klebsiella pneumoniae, and this is again a huge plasmid which we identified, and this was a plasmid which was carrying both resistance genes and it was also carrying virulence genes. So if you have such plasmids which have both resistance and virulence, you know how difficult it is to treat such bacteria which cause infection. So this is just to give you a glimpse of the different techniques that we use. The simple distribution test is an essential part of any work that you do in antimicrobial resistance. Then you have the identification of genes by different PCRs and sequencing of those genes. And then you can also do whole genome sequencing to compare strains, to compare resistant genes, and look for different signature types. So again, giving you a glimpse of some work that we had done on septicemic E. coli and Klebsiella pneumoniae, where we looked into the mechanisms of resistance and the mode of transmission of these genes, and we published this work in PLOS, where we shared our experience of carbapenem resistance in Enterobacteriaceae, which was causing neonatal septicemia because of the gene that I already mentioned, the NDM1. This was a work on another very important bug, the Acinetobacter, which actually is a major cause of mortality and morbidity in neonates, or not only in neonates, in any intensive care unit. This is one bug which plays a very important role. So we were doing some work on Acinetobacter, where we compared these strains. We looked into the different types of strains we had by a technique called the pulse field gel electrophoresis. And we also carried out work on the efflux pumps of these genes, and we published the work in Frontiers in Microbiology, carbapenem resistance in Acinetobacter, again causing neonatal infections. This is a work that we published very lately in 2022. And this was again on drugs, a drug called colistin, which is generally used only in very serious infections. And we were studying this mode of the mechanism of this mechanism of drug resistance towards colistin. And there are different mechanisms, as I have already mentioned. They can be pumps, they can be other mechanisms of resistance. In case of colistin, where you have different, what is called the two-component system, where these get activated and finally will bring about changes in the cell membrane, due to which the bacteria becomes resistant to that particular drug. And then what we did was we compared our strains to a collection of these similar strains across the globe. And we found that our strains were very diverse, but the mutations that we were getting in our strains were also seen in other global strains. And you can also see a similarity in the different antibiotic resistant genes present in these strains. And here we had actually carried out whole genome sequencing of our strains to compare them with other strains. And why is it important to do that? Why is it important? The importance is because you need to understand whether the strains that you are seeing locally have spread across the globe, or whether you have certain clones which are called epidemic clones, which can actually cause infections in different parts of the world. So we also carried out a study with around 6 other low middle income countries. So there were some countries from Africa, and there was India, Pakistan, Bangladesh working together. So this was the study which we carried out, where we again looked into the burden of resistance, where we characterized the antimicrobial resistant gram-negative bacteria, and we published this work in Nature Microbiology, and we also had a follow-up publication in Lancet, where we showed the different nuances of drug resistance. We also do a lot of work on the genetic environment of antimicrobial resistant genes. Again, why do we need to do that? We need to do that because apart from the gene, we need to understand the environment in which the gene is present, because the environment of the gene, whether it is present within mobile genetic elements such as transposons, whether it is present in plasmids, and there are certain transposons which can move, spread across different species very quickly, and we also have plasmids, certain plasmids which have the capability to move across different species. So that is the reason why we do a lot of work looking into the environment of these antimicrobial resistant genes. We also did a very important work on the outer membrane vesicles, where we actually showed how the outer membrane vesicles, these are small sac-like membranes which can actually act as a vehicle for the spread of antibiotic resistant genes. So this was again another work where we showed that we generally know that the spread of resistance happens from one bacteria to the other via conjugation, or it can happen through phages, which is called transduction. But this was another novel way in which the resistance to certain genes were getting spread. So this was more or less giving you a glimpse of the different kind of work that we do. And these are some of the publications, some important publications. But all of this is to emphasize that antibiotic resistance is a huge problem, particularly in neonates, particularly in a country like India. Now, just a quote by Wallace Stevens before I move into the next part of my talk, where he said that in the sum of the parts, there are only the parts. The world must be measured by I. The point is that when you talk about the problem of antibiotic resistance, what you have now had an idea of are different parts of the problem. So these are little parts of the whole problem. The problem itself needs to be looked into in a much more holistic way if we really want to bring a solution to the issue. And when we look into this problem, we will, all that I have been speaking about, the work that we do is primarily focused on hospital infection. But it is not the hospital, it is not the clinicians who play a major role. We also have a lot of antibiotic resistance in animals, in the environment, and all of this play an extremely important role in the spread of resistance. So the hospital plays its role, and that is quite understandable. But there's a lot of, like, particularly in poultry, in fish, the use of antibiotics in poultry farms have been very rampant until a few years ago, where actually colistin, which is one of the important drugs for humans, was getting used in farms as animal fodder or poultry fodder. So animals, if this happens, then they contribute a lot to antibiotic resistance. So the resistant bacteria can enter the food chain. Then you also have all of this having an interaction with the environment. And there is also the pharmaceutical industry, which plays an important role. And you all know that India is one of the largest hubs of pharmaceutical production. And this, the accidental or intentional release of antibiotics from the plants to the environment, and then from the environment to the animals, and then to the humans, all of this is needs to be tackled at one go. And that is the reason why the term One Health has been coined now, because the problem of resistance was initially thought to be a problem which was limited to the hospitals and the clinicians and the use of drugs by the humans. But now we see that there are different angles to the whole problem. Now, so what are we now doing? Apart from the scientific research that is going on, what else is happening across the world? The first important thing, or the most important role that has been played is by the World Health Organization. And you as students are all aware that the World Health Organization now has a global action plan for antibiotic resistance. And each of the countries have been asked to draw up their national action plan. And this national action plan that you see here is that of India. So India has a national action plan which aligns to the global plan. And this is the plan where we start with, or we parallelly do each of these, where we improve awareness and understanding of AMR through effective communication, education, and training. This plays an extremely important role because the more people are aware about the problem, at each level this can be tackled. The others are, of course, strengthened knowledge and evidence, which comes through surveillance, which comes through research. And a part of that is the kind of work that we do, where we carry out different work in different hospitals across India, across the globe, where we create evidence that this is a big problem. Then you reduce the incidence of infection through effective infection control measures. When I talked about the study in the district hospital in Shuri, I talked about an important role of infection control, because as we know, if you have good infection control practices, then the rate of infection will be low and the use of antibiotics will also go down. Apart from that, you need to optimize the use of antimicrobial agents in health, animal, and food. All of that I've explained earlier. And promote investments for AMR activities, research, and innovation, and finally strengthen India's leadership on it. And this strengthening of leadership has to happen across different sectors, and a very important role is played by many different organizations, and particularly people who make policies. So we need to, as scientists, as clinicians, we have limited access to making policies, but we can, through our work, influence the policy that we have about the use of antibiotics. Now, this particular slide is a slide which is primarily focused on the kind of work that we can do in research around antimicrobial resistance. So there can be newer drugs that can be discovered. A few of the drugs are being discovered, but the pipeline has really been dry for the last few years because there are no investors for the amount of research and the money that goes into the discovery of new antibiotics. Because this is a drug which will cure you in 5 days, and then again there is a problem of resistance. Again, these drugs will not be working, and there are drugs which people take for lifelong. A drug like a drug for diabetes will be used for by a person for years, whereas an antibiotic really is used for 5 to 7 days, and in some cases, in very debilitated patients, in neonates, you can have long-term usage, but that too only a few months maybe. So the discovery pipeline has been really, really dry. A lot of work is being done on the epidemiology part. Then NICED is also trying to bring in a repository of antimicrobial resistant pathogens. So you will have a storehouse of these pathogens which can be further used for research and also for drug development. There's a lot of work being carried out on the different mechanisms of resistance. As I said, the bacteria come up with newer resistance every day. So you need to actually, as we heard in one of our slides earlier, we need to stay ahead of the curve. If you really need to stay ahead of the curve, this is where we need to invest in, but we also need to bring in a lot of changes in the policy. So there can be a lot of work again on the genomes, because this platform has given us immense access to bacterial genome data, which can be used in a very important way later on. Then there are diagnostics, which also play a very important role. The quicker you diagnose an infection, the correct drug you will be using, and the correct way in which patients will be treated. this is a slide which again shows that the problem of antibiotic resistance is extremely difficult to solve. There are several initiatives which are getting carried out across the world. There are the pipeline reinforcement for different things are being carried out. There are certain initiatives apart from WHO. There are other organizations which are also playing a very important role in creating data, then pushing for policy changes. But there are also other ways in which this change can be reversed if we do not really take care at the right time. I'm nearly at the end of my talk, and this is Joshua Lederberg, the Nobel laureate, wrote that the future of humanity and microbes will likely evolve as episodes of our wits versus their genes. You know that their genes have already fooled us or have already stayed ahead of us in many different ways. But now we need to use our wit to tackle this problem. And as we know, if there is a will, there is always a way. But we need to really resolve to stop inappropriate antibiotic use in humans and in animals, and we should look for alternatives. And how can we do that? So what is the way that we can do? Of course, scientific advances are an important thing, but we need to push for newer policies. We need to push for more awareness about this particular problem. And I will now finish my talk. This is my team, and most of the work that you have seen has been carried out by some of my senior students here who are not in this picture. This is a more recent picture of the students in the lab. But most of the work that I have shown you, like I have discussed here, is a work carried out by my students, Shubhosri, Shashwati, Shomlata, Shubhankar. Shormi is here in the picture. Amrita is here. Some of their work has been presented here for you. So I would end my talk with this, and I'll be happy to answer questions.
Thank you, ma'am. Anyone having any questions?
Okay. No, I'm comfortable standing. It's fine.
Anyone having any question? I have a question from my end. In your slide, I was seeing that you have mentioned that I think in the beginning about the mechanism of how antibiotics works and how antibiotic resistance works, where you have said that the pouring channels used to allow the antibiotic entry, which used to get its numbers increased. That's why the antibiotic is not being able to enter. And so, ma'am, as for my information I'm having, that pouring channels are basically allowing the water soluble molecules to come inside. So if the antibiotic is highly water soluble, they only need to send out, or it's for
no, it is many antibiotics enter the bacterial cell through the porins, and there are multiple porins. Now, if your question is, madam, if they lose the porins, then how do other molecules enter? The point is that there are several porins. Each bacteria species will have certain porins. They will, of course, they are clever enough not to lose all of them. But what they do is, suppose in E. coli you have, in Klebsiella you have the OMP, outer membrane protein 36, 33, all of these are present. There are OMP-A, several of them. Now, in certain cases, what we have seen in clinical isolates is that there is a mutation in, suppose, the OMP36. So because of that extensive mutation, the porin gene doesn't express. So you don't have that particular porin, due to which there is a reduced entry of the antibiotic. So in most cases, there are multiple mechanisms working together. There is a reduced entry of that antibiotic. Along with that, you may also have an hydrolyzing enzyme like an ESBL or something, which can hydrolyze the antibiotic also. So both of these mechanisms are working together. So that's
one is big block and other is one other.
No, that the reduction of porin does really bring down the amount of antibiotics that enters. So the little amount that is entering then can get hydrolyzed and then you finally have resistance. In certain cases, the porin can, like suppose in case of certain bacteria, you have the loss of multiple porins also. So that creates a bigger problem.
Another question that you have mentioned about a New Delhi metal beta lactam. So how is it different than previously? Because this is the first information I've heard about this enzyme. So we used to know the beta lactam enzyme, which actually creating the resistance about the
against the beta lactam drugs.
Right. So how these two enzymes are different?
Yeah. So see, if you just look up New Delhi metallo-beta-lactamase, this was an enzyme. The point of time when we were carrying out the work in Shuri, it was identified at that time, and it was named New Delhi metallo-beta-lactamase because it was identified from a patient who got treated in Delhi, New Delhi, went off to Sweden, and then they identified a bug which was having that particular resistance gene. Now, to come to your question, is that this particular enzyme can hydrolyze just like the beta-lactam, the extended spectrum beta-lactamases. This also can hydrolyze a number of beta-lactam drugs, but it also has the capability to hydrolyze the carbapenems. The ESBLs will not hydrolyze the carbapenems. Carbapenems are also beta-lactam drugs, but ESBLs will hydrolyze the cephalosporins, which are the beta-lactam drugs. But NDM has a capability not only to hydrolyze all the other cephalosporins, but it also can hydrolyze the carbapenems. Because of its extensive activity, this has now become a huge problem. And this particular, as when I was mentioning the history, in the whole issue of antibiotic resistance, awareness of this problem, I think the world woke up to the problem again after several years in 2009 and 10, when this particular drug, and there was a huge furor across India also about why this enzyme was named after New Delhi, and that this was a way in which you are showing India in a poor light, because India is a very important hub for medical tourism. You know, patients come here from different countries across the globe because the treatment is pretty affordable here still now, and you also have good treatment. So there was a huge furor, but with this drug, with this enzyme, I think the world nearly started realizing that the situation is really, really bad. And if you now look up into, I haven't shown, there are slides which have shown that NDM has been found from nearly all parts of the world. And if you see places where it is not present, it's because nobody has looked for it. That is the reason. So it has been so widespread. So it's like around 10, 12 years now, and it has spread all across. And that is the problem of resistance. And this being an enzyme which is actually hydrolyzing carbapenems. Carbapenem is a drug which is used extensively in ICUs because I would say it has been misused for several years in ICUs, and now you are having drugs which are resistant. Yeah.
Thank you, ma'am.
Sorry, ma'am, I was little bit sorry. I couldn't follow whole the lecture, but as we were talking on this one, carbapenems, right now we were looking about the antibiotic resistance genes, ARGs, to locate those parts. So how do you think the gene regulation can modulate this kind of carbapenems resistance? What can be the major pathway that could be through this genetic modulation? What is the basic means? It's not in your topic, but just for my interest.
No, I couldn't get your question completely.
So the ARGs now they are
Yeah, there are so many ARGs.
yes. So the which particular ARGs are directly connected with the carbapenem resistance?
Okay. One is, of course, ARG is antibiotic resistant genes.
Yes.
For carbapenem resistance, one is the NDM, which I've already mentioned. I did not go into details, but there are other genes like, suppose, the KPC, which is the Klebsiella pneumoniae carbapenemase. So that also can hydrolyze the carbapenems. Then you have a group of enzymes called the oxacylinase. There are some which cannot hydrolyze the carbapenems, but some of the oxacylinase can hydrolyze the carbapenems. So you have OXA48 in Klebsiella pneumoniae, which can hydrolyze carbapenems. So these are the ARGs. And in case of Acinetobacter, you also have OXA23. You have OXA58. They can also hydrolyze. These are the ARGs which can hydrolyze.
And would we select any pathway where we can resist these hydrolyzations? No, not yet.
No, the primary, these are all enzymes. These genes are all encoding the enzymes. So the enzymes, if you want to, the major focus has been on actually focusing on the plasmids, which are being the vehicle for this transfer. And you can also have a lot of carbapenemase inhibitors are already in the market. You know that sulbactam is there. You have now latest, I think, avibactam is there. Piperacillin tazobactam is there. All of these can actually act as inhibitors of these carbapenemase. But apart from that, the only way you can focus, or if you think you can really bring about a quick change, is by focusing on the plasmids, the spread of the plasmid. But that also has their own limitations. The reason being that these are genes which are placed on different plasmids. They are moving in different plasmids. So you need to target it.
So like the transposons now they are moving, jumping genes. So we need, so they are moving like
Yeah.
Okay. Yeah, that's more challenge now. This happens in a larger scale.
The challenge actually is that if you reduce the use of antibiotics, then you slowly see that the bacteria doesn't really maintain these plasmids. Of course, it's a fitness cost on the bacteria itself. So they slowly lose the plasmids, because why would they keep something there which is not required? It is only with the constant use of antibiotics.
So with the gap, we can again increase the sensitivity.
Yes,
profile of the antibiotics.
yes, and in many ICUs, I have seen clinicians will use a process where they call rotation of an antibiotic. So they just stop using a particular antibiotic in that unit. When they see that the resistance is getting higher, they stop using it, and then they bring in other antibiotics, and then slowly, maybe after a few years again, they start using it. So that's a way of it.
Thank you, ma'am, so much. Thank you so much.
Sorry, I was not part of your talk, but thank you for the So there's a lot of buzz about intestinal microbiota and how it affects AMR. Somehow enriching it can be prevent antimicrobial resistance, but then again there are some literature that shows that resistance can be transferred to industrial antibiotics. So what is the
Yeah, we have, this is again a very interesting part of antimicrobial resistance, and we have already done some work, primarily because we work on neonates. We have looked into the transmission of these genes from the mother to the neonate. So you take pregnant mothers, and we did a study which was called the Barnard study, where we looked into the transmission. And when we were looking into the transmission, the samples that we had collected from the mothers were the fecal sample. From the babies also we carried it. The carriage of resistance genes in mothers is extremely high. Extremely high. It's unimaginable for certain antibiotics or certain genes like CTXN, these cefotaximase, which can hydrolyze, which is a ESBL, which can hydrolyze many of the beta-lactam drugs, is present in 60 to 65% of those. Yes, and as you said, do these genes spread within the flora? Of course they spread. It's a huge place where it spreads. But as I said, if you are not taking inappropriate antibiotics, if you minimize that, of course, this will slowly be replaced. And also what we need to understand when I discussed was the entry of these genes through the food chain. The poultry farms have been using a lot of antibiotics in their fodder, which again gave rise to the resistant bacteria, which we are using in our daily life. That enters us. So that also in mothers, that really antibiotic resistance has been high. In neonates, yes, for neonates, when a neonate is hospitalized for a long time, you can very quickly see the flora changing, like from a very susceptible flora to becoming more and more resistant, because once they stay in the hospital for a long time, the hospital environment actually triggers that. So apart from that, but the mothers, when we looked into them, they were healthy pregnant mothers. So probably they have these genes because it is already in our food chain, has entered our system. Yes, it's pretty scary.
Thank you, ma'am. Any more questions from the audience?
That side
So,
from the PMIT students, anyone?
who is that? Any?
Even if you don't have any questions now, I hope you can go back and do a Google search and see how big this problem is and how across the globe everybody is trying to deal with this problem, because we have dealt with a problem like COVID a few years back, and antimicrobial resistance is being considered as a tsunami. So it is happening quietly. You only realize it when you are in a hospital, when you have a infection, you know, and you do not get treated. When you see your loved ones in elderly, particularly elderly patients in hospitals, going there for surgeries, coming back with antibiotic resistant infections and not getting treated. When you know somebody who has had a recurrent urinary tract infection which is not getting treated, you will only know at that point of time that this is a problem that we cannot ignore. And I really want this as my talk. Don't go into the nitty-gritty of the research, but try to understand the enormity of the whole problem and try to become more and more aware about this, And NICED really will play a very important part in the community, trying to raise awareness about antibiotic resistance, because really see that if we do not, this will become a huge problem for us. And I have really been working with the neonatal unit. This is the IPGMER SSKM hospital. The unit is one of the best units in Kolkata. Their infection, yes.
Take the mic.
ম্যাম আপনি বললেন না যে যদি হসপিটালাইজ থাকে কিছুদিন, তো সেক্ষেত্রে ওর অ্যান্টিমাইক্রোবিয়াল রেজিস্ট্যান্সটা ইনক্রিজ হয়ে যায়। তো তার জন্য কি করতে হবে যেন সেই জিনিসটা না হয়? কারণ একটা বেবির পক্ষে একটা জাস্ট বেবি, তার ইমিউনিটি একদমই নেই, যে মাদার্স থেকে ডেভেলপ করবে। এবার সেক্ষেত্রে আমরা দেখছি যে ওর অ্যান্টিমাইক্রোবিয়াল প্রপার্টি রেজিস্ট্যান্সটা একদমই হাই হয়ে যাচ্ছে। তাহলে ওর কি করণীয়, ওখানে কি করতে পারে?
না, করতে তো বেবি পারবে না, করতে পারবে অ্যাকচুয়ালি হসপিটালে। এবং এরা কিন্তু এই ইউনিটটা পার্টিকুলারলি আমি যেটা বলার চেষ্টা করছিলাম, এই ইউনিটটা কিন্তু এক্সট্রিমলি ওদের ইনফেকশন কন্ট্রোল প্র্যাকটিসেস আর ওদের ইনফেকশন কন্ট্রোল প্র্যাকটিসেস কিন্তু ভীষণ ভালো, মানে এত ভালো যে অ্যাকচুয়ালি খুবই কম, মানে 5% এর কম ওদের ইনফেকশন রেট। I have now spoken mainly রেজিস্ট্যান্সটাকে ফোকাস করে, কিন্তু ওদের ইনফেকশন রেটটাই খুব কম। তুমি যেটা জিজ্ঞেস করলে না যে কেন কম, এই হসপিটালেও কিন্তু প্রচুর চেঞ্জেস করা হয়েছে 2010 অ্যারাউন্ড। কিন্তু এই যখন এই এনডিএম এর ব্যাপারটা শুরু হলো, তখন থেকে রিয়েলাইজেশন হলো যে, আগেও ছিল কিন্তু তখন থেকে রিয়েলাইজেশন হলো যে ইনফেকশন কন্ট্রোল প্র্যাকটিসটা যদি যত ভালো হবে তত কিন্তু ইনফেকশন কম হবে। দেখো বাচ্চারা তো এমনি হেলদি বেবিকে কখনোই হসপিটালাইজ বেশি দিন করা হয় না, তোমার 2 দিন 3 দিন ম্যাক্সিমাম বাচ্চাকে কিন্তু মানে তোমার ডিসচার্জ করা হয়। হ্যাঁ, প্রিম্যাচিওর বেবি এরা থাকে তো, দে টেক আটমোস্ট কেয়ার। ওই ইনফেকশন তোমার ওই হ্যান্ড ওয়াশ, তোমার ক্লোরোহেক্সিডিন রাব, তোমার যখনই নার্স বা কেয়ার গিভার বাচ্চাকে হ্যান্ডেল করছে, তারা কেয়ার নিতে হবে যে যাতে তোমার তাদের হাত থেকে কোনো রেজিস্ট্যান্ট ব্যাকটেরিয়া, মানে ইনফেকশন কন্ট্রোল ইজ দি বেস্ট ওয়ে টু প্রিভেন্ট। এন্ড এরকম এই হসপিটালেই আমি বলছি, এদের বেড টু বেড যে ডিস্টেন্সেস, যে বাচ্চাদের কটটা, আগে এদের ইউনিটটা অনেক ছোট ছিল, এরা বেড টু বেড ডিস্টেন্সেস বাড়ালো, এরা বড় জায়গায় স্প্রেড আউট করে দিল, অনেক ইনফেকশন রেট কিন্তু কমে গেল। মানে ওই যে একটা ছোট জায়গায় অনেক পেশেন্ট যখন ক্লাটার থাকে, অবভিয়াসলি ইনফেকশন কন্ট্রোল প্র্যাকটিসেস ভেরি পুয়ার। কিন্তু যেই বাচ্চাগুলোকে কটগুলো অনেক স্প্রেড আউট হয়ে গেল, অনেক হলো, ইনফেকশন কন্ট্রোল প্র্যাকটিসেস আরো বেশি মানে রিএনফোর্সড হলো, তখন কিন্তু এমআর এর রেটস কমে গেল। এখানে আমি যেটা দেখাচ্ছি, কারণ এই টোটাল কত বাচ্চা বর্ন হচ্ছে, only 5% is having sepsis, which is a very very nominal rate. তার মধ্যে অফকোর্স ওই রেজিস্ট্যান্ট ব্যাকটেরিয়া গুলো কিন্তু প্রিডমিনেট করে, বিকজ দ্যাট ওই ব্যাকটেরিয়া গুলোই তো সার্ভাইভ করে না ইন দ্য প্রেজেন্স অফ অ্যান্টিবায়োটিক্স। মাদার থেকে ওই হ্যান্ড, মানে মাদারের দেখো ডিউরিং দ্য প্রসেস অফ ডেলিভারি সেটা তো হবেই, তাই না? মানে সেটা তো বার্থ ক্যানালের মধ্যে দিয়ে অনেক সময় হয়। বেবির যদি ইমিউন সিস্টেম ঠিক থাকে তাহলে সে আপনি আপনি সেটাকে রেজিস্ট করতে পারে, কিন্তু যখন সেটা পারে না তখন সেপসিস তো অফকোর্স হয়। আর ডিউরিং কেয়ার গিভিং ও সেটা হয়, কিন্তু healthy baby কিন্তু is not so prone to the infection. It is only these low birth weight babies যাদের immune system টা অত develop করেনি তাদের।
Thank you so much.
I'll request you to stay back for a minute. So I'll request the session chair, Dr. Ritu Khara, to felicitate our speaker. Thank you, ma'am, for your time. Thank you. It's a small token of appreciation from our department. Thank you so much, ma'am. We are very much thankful to have you. Thank you, ma'am. I hope all the participants are actively participating in our session. And I'm hoping that all of you are paying attention and learning a lot of new information from the various speakers from the various aspects of science. They have come here and sharing their experience of their research, of their knowledge with us, because the conference is only for upskill our knowledge from the place where we are now to get a new more information to get, because we all are, most of us are going to be researchers. We are on somehow involved in different segments of research, maybe that is a little scale or a large scale research. So I'm sure the information our speakers are providing is being very much helpful to all of you. So without making a late, I would like to announce that our second speaker of the day for the second day of the conference is already with us, Dr. Sugata Banerjee. So I'll request her to please come on the stage, sir. Thank you so much. I'll request Dr. Tapan Kumar Shaw, the professor of Art Department of Pharmaceutical Technology, to welcome him. So, thank you.
Good morning, everyone.
Good morning, sir.
Dr. Sugata Banerjee from his busy schedule, he has came here and delivered his lecture on the metabolic disorders associated with cognitive decline. So, which is very much the related topic to our conference, Advanced Therapeutics for Life-Threatening and Chronic Disease. So let me introduce Dr. Sugata Banerjee first. Sugata Banerjee. So Dr. Sugata Banerjee presently working as Dean in charge, and he is serving as associate professor at NIPAR Kolkata. He has more than 12 years of teaching and 20 years of research experience. He has completed his PhD in pharmacology and experimental neurosciences from University of Nebraska Medical Center, USA, and worked as postdoctoral fellow University of California, San Diego, USA. His current research interest includes understanding of molecular disease or molecular pathway associated with metabolic disorders, and also associated with CNS complications, including various demential disease and cognitive impairment. the work group of Dr. Banerjee is trying to understand the role of gut bacteria as well in memory changes. And they have published many papers and try to find out establish this metabolic disorder, association of metabolic disorder with cognitive decline by various in vitro and in vivo animal studies. He has guided 3 PhD students and 30 master students, and he has more than 50 international publications in his credential, and also a number of book chapters he has published in many reputed journals. And also he has handled one extramural project of ₹1 crore. So this conference topic, which is advanced therapeutics for life-threatening and chronic disease. So I think this topic of discussion of Dr. Banerjee, metabolic disorder, is much related and very much significant. Now Dr. Banerjee will deliver his lecture on this topic. Sir, the session is yours. Thank you.
Good afternoon, everyone. Fellow students, faculty, and other dignitaries. So basically, my focus area is metabolic disorder associated brain changes. So I mainly focus on cognitive changes. However, there is a huge literature showing that how metabolic disorders can affect depression and other psychological complications. So here mostly I will be talking about the work we are doing in our lab, and basically we are trying to determine certain intervention strategies so that individuals, so here mostly we will talk about animal studies, but the main implication is how to reduce the chances of, by cognitive changes, we mean here mostly we are talking about memory changes or memory decline, and what are the ways we can prevent this. So this is from the literature. You can see that chances of Alzheimer's disease increases by 2 to 4 folds in individuals having diabetes, while various factors are involved for this, like you can see there.
No, I think.
So here you can see that various factors like insulin resistance, obesity, cardiovascular complications, all these risk factors adds up to the development of cognitive impairment or memory decline. And you are more susceptible to Alzheimer's if you are having diabetes. And that is a very well established fact now. So now how to prevent it? So various mechanisms has been proposed how diabetes is being linked with the development of cognitive impairment. Like you have hyperglycemia, amyloid beta accumulation. Amyloid beta you might have heard for the students that these are the one of the major proteins, accumulation of these proteins leads to Alzheimer's. So it has also been shown that individuals suffering from long-term diabetes, they have higher levels of beta plaque in their brain. But this is only one of the mechanisms. Then you have oxidative damage, which is again a very common factor for any neurodegenerative disorder. And this oxidative damage actually leads to inflammation in the brain, which ultimately may manifest as a change in your cognition. And acetylcholine, as you again, most of you pharmacology students and others in the audience know that this is one of the major neurotransmitters associated with memory. So, and again, the same thing has been shown in patients with long-term diabetes that there is decreased level of acetylcholine. And probably you also know that in Alzheimer's patients, the current therapy revolves around acetylcholine and acetylcholinesterase. So somehow if you can, that's the cholinergic hypothesis. If you somehow can increase the level of acetylcholine, you will prevent the development of cognitive effects. Or you can sort of partially reverse Alzheimer's like pathologies. So these are the current hypotheses regarding metabolic. So here mostly we are focusing on type 2 diabetes because our work revolves around type 2 diabetes. We are not looking at metabolic syndrome or hypercholesteremia kind of diseases. So here you can see the various mechanisms that are linking this both in human as well as in animal studies. They have shown that this insulin degrading enzyme. So this insulin degrading enzyme has been shown in a lot of studies that it also helps in the degradation of amyloid beta, which I will show you in the next slide. And this altered insulin signaling because of this change in insulin degrading enzyme actually can affect your insulin receptors that these are your glucose transporters. So there is internalization of glucose transporters, and the whole insulin, the pathway in the brain gets affected. And the secondary consequences for that, again, you can see. Can you hear? So here you can see probably this again as a secondary effect of this, you have here this oxidative stress, and then this activation of microglial cells, and then your NF-kappa pathways. These are basically your receptors, yes-20 NF-alpha receptors. So it basically affects the inflammatory cascade in the brain, and as a result of which, again, you have neurodegeneration, and ultimately leading to cognitive decline. And as you can see again in the next slide, you will see it's just not the neurons alone, but your astrocytes, microglia, all are involved. So this is one interesting hypothesis, or rather, people have been working on this. So metformin, which is a very common hypoglycemic drug, the first time for diabetes, it has shown neuroprotective potentials. So I'm talking about metformin because in our work also we have used this drug along with some other interventions. And here you can see basically how it affects the release of neurotransmitters, both your pyramidal neurons as well as these are the interneurons or the inhibitory neurons or the GABAergic neurons. So how it is affecting the release of neurotransmitters. While these are the astroglial cells, so if you are not aware that reactive astrogliosis, or what we call as gliosis, so astrocytes are supporting cells in the brain. They maintain the homeostasis, they protect the neurons, but if they are activated because of this inflammatory cascades that are upregulated, again in diabetic conditions in the brain, they may also affect the well-being of these neurons. So again, it has been shown that this metformin affects both neurons as well as astrocytes. So as a result of which, it suppresses neuroinflammation as well as it modulates the release of neurotransmitters. And if you maybe have studied a little bit about learning and memory, so you know the role of NMDA receptors, AMPA receptors. So these receptors are very important for your synaptic plasticity, or rather your brain is a very plastic organ, so it changes, can be modulated. It is not like any other organs. So that is how your memory is consolidated by activation of the NMDA receptors, AMPA receptors, and glutamate is a primary neurotransmitter for this. So somehow if you can modulate the levels of glutamate, if you activate it too much, it is not good. It will lead to toxicity. If you don't activate it at all, it is also not good because you will not be able to consolidate your memory. So memory formation will not happen. So, and metformin has been shown, though it is established anti-diabetic drug, it has been shown to have all these activities, which is a good news for people who are having diabetes and on metformin. So they become less susceptible to development of cognitive changes at the older age. Like I mentioned, all these changes happen after a certain age because age is a major factor here. So more than 65 a person is more likely, and having prolonged diabetes is more likely to have some decrease in memory processing. So at a younger age, it may not be that relevant. So these are some of the work by our group and others where people have shown that so this animal model of type 2 diabetes is the established model HFDS is chemically induced diabetes, and we primarily work on this model. There are also some genetic models, but we don't have those. And these animals tend to develop cognitive decline. So after 8 to 10 weeks, or sometimes little more of diabetes in these animals, they tend to perform less well in memory tasks. I will show you the memory tasks there. And the primary mechanisms for that we studied, and it looked like it's more to do with neuroinflammatory changes as well as oxidative changes in the brain. And there is a clear literature on role of exercise on neurogenesis. So again, I'm mentioning this because in my study, you will see that we subjected the animals to exercise as a natural intervention therapy, as well as we administered them with metformin. They were on metformin, and that actually shows some promise. So the main objective of our work was to determine the effect of exercise and metformin in this SD rats who has developed prolonged diabetes, as well as these rats have developed some cognitive deficits at the same time. So this is basically the study design that we follow. So we give a single dose of streptozotocin, and the animals are already on high-fat diet. So in high-fat diet, basically it has more than 70% of the calorie comes from fat. So and then these animals develop diabetes on the 4th or 5th week. And now if we continue this, for up to 13th week, or rather around 10th week, they develop, if you don't treat them, they develop cognitive changes, or they show, compared to a control animal, they show, and we have seen that in all our previous studies, they show less memory performance. And in this particular study, basically, we have subjected them to treadmill exercise and as well as administering them metformin right from 1st to the 12th week. Around the 13th week, 12th to 13th week, we do the behavioral studies. So I will explain you the behavioral studies. And then we do other biochemical, histopathological, and stuff. So this is the aerobic exercise platform we use for. Again, this project is ICMR funded project, so we didn't have problem procuring the things. So this is basically animal trade, which is way more expensive than a human trade. So this was actually from market, so other companies are also making it. So it costs like 40,000-50,000, which is much more expensive than a normal trade, because it is very specialized and it has all this. You can control the speed, you can control the inclinations, and you can have basically here you have this animal platforms here. And so the anyway, it is not easy for just you don't put an animal there and they start running. So a lot of initial acclimatization and training is required till you get them going, because animals are also lazy like humans. They don't want to move around too much. So here one feature of this instrument is it has a shock grid. A mild shock grid is there. So if it does not run, you give them a mild shock. So now they start running. Otherwise, they will stand there. They will not run. And so it kind of acts like you don't have to give them shock all the time. Then they understand. Like when I am put on there, and if I don't run, they will give us the shock. So they start running. Okay. So that is how its instrument kind of works. So first, week 1 and 2, we give them 30 minutes exercise or running time. Third and fourth week 45 minutes. And this is the established protocol. They are not discovering it. So week 5 to 12, they are running for 60 minutes every day. 5 days. Okay. And these are some of the behavior instruments probably you are accustomed with also in pharmacology. And these are pretty regular. So they have the Y-nails, which basically here, if the animal is going in all the 3 arms, it is normal. If it is repeating, coming back to the same place, we call it incorrect readings, or rather the animal is somehow not normal. Then you have the passive avoidance test, which checks again, this is the fear conditioning test. Again, I'm explaining this more for the students at the back. So to make you interested into this behavioral pharmacology, this we call behavioral pharmacology. So here basically we give a fear conditioning, and so in one side there is a shock treat, the other side is just a normal enclosure. So once the animal goes to the other side, it gets the shock, and then it knows whether to go there or not. If the animal is not having a proper memory, it will forget. So it will keep on going to the shock zone. Even if you don't give the shock, usually in this experiment, initially you give the shock, after that you don't give the shock. But a normal animal will not go to the zone where it received the shock maybe 2 days back. But the other animal will keep on going. So we said that the animal is memory impaired if it keeps on going to the shock zone all the time. Novel object recognition, again a very simple test. Both for human as well as animals, we have a tendency to explore new things. So that is basically used in this particular test. So if the animal is not exploring new things, that means probably it is not remembering. So anything you put the same thing again, it does not remember it as a new thing. It will remember, okay, maybe this is a new. But if a normal animal will remember, okay, they had already produced this. This is nothing new. So I will not explore this. I will explore something that maybe this one you get later. So I will explore that more. So this preference or this exploration index we call tells you whether the animal is normal or it has some cognitive deficit. So these are the three parameters mostly we use. We can also do a Morris-Potter mess, but we don't have a Morris-Potter mess currently. And all this is not manually done. These are all video tracked. So there is no chances of a biasness here. So everything is recorded, and this, which path it is going, you can visualize in this. We call it a video tracking software, and same thing here also. So next we did some biochemical studies like histopathology, histochemistry, and Western blot. So that I will show you the data, because this technique anyway you can read anywhere. It's pretty well established. So initially the animals developed diabetes as well as hyperlipidemia. The main reason because we are giving them high fat diet. So they will not only develop diabetes, they will also develop high lipid profile. So blood glucose levels are pretty high. And we use this model because this is the established model for type 2 diabetes. That's why we give a very low dose of streptozotocin. If you give a very high dose, you will develop type 1 diabetes. But type 2 diabetes is more prevalent in the population. Type 1 is more of a genetic thing. And so the patients that probably a clinic receives having diabetes more are of type 2. Since our current lifestyle is kind of high fat diet kind of lifestyle. So chances of developing type 2 is very high. So anyway, they develop pretty high blood glucose, triglyceride, cholesterol, as well as change in the HDL. So these are some of the rather after the intervention, you can see here the various groups. So you have the control, you have your disease group, you have after exercise, and you have exercise and metformin. So here what we saw is in most of the cases, here you see a significant difference, but this not too much. So exercise is doing a major role here. Metformin kind of increases it a little bit, but the major change you see here between this high fat ST group or your diabetic group and the intervention is exercise is actually playing a major role in partly reversing this. So YMS test, you can see here the spontaneous alterations is good. So there's a reduction in spontaneous alteration which goes back, goes to normal. Again, discrimination index, this is for your like how you discriminate between two objects, whether it's novel or it's object you are giving the animal all the time. Then you have your transfer latency. This is basically done in your YMS test. In your passive avoidance test. So transfer latency is basically whether it is going to the shock chamber or it is, or how long it is going to the shock chamber versus not. So this axis you can see is in seconds. So there's a reduction in this diabetic animals, whereas it's not exactly same as the control, but it's improving quite a bit. So these are the biochemical studies, because in behavioral work initially, and again this is for the ethics purpose also, again for the students, you should never sacrifice an animal or never try not to disturb it physically or abuse it physically unless you see some changes in behavioral studies. So only the animals which are showing some alterations, then you go to collection of their blood or collection of their tissue. Otherwise, your institution animal ethics committee will never approve your product. So this is a norm and you should follow it. So here basically we looked at acetylcholinesterase levels, which is a very standard thing for any memory study. So whether there is a change in acetylcholinesterase level, basically you know that it is an enzyme taking your acetylcholine. So obviously we correlate this with our acetylcholine levels. So if there is an increase in acetylcholinesterase, there will be a corresponding decrease in acetylcholine. So we look at their levels and we see here the huge increase in acetylcholinesterase, which actually never came back to the normal, but it to a certain extent it was reduced. So you can see we are unable to totally reverse it, but somehow improving the conditions by both exercise as well as metformin exercise. And then we looked at the inflammatory conditions in the brain. So whether there is inflammation in the brain, we look at this, and these are all hippocampal homogenetes. So TNF alpha, interleukin 1 beta, MCP1. So all these kind of correlate with inflammation in the brain. More inflammation in the brain, more you will have the levels of these cytokines and chemokines. And we saw here a reduction in all these cytokines and chemokines. As well as we did not do much with the oxidative stress, but only with the GSH, and it showed a trend, but not too much of a improvement here. So it was more towards the inflammatory markers. The next thing we looked at is what is happening with the hippocampal neurons. Whether there is a loss in hippocampal neuron, and probably all of you might be knowing that for working memory, hippocampus plays a major role. Again, there are other memories for which you may have, like emotional memory, you have amygdala, which is a major organ or major part of the brain. But for working memory, hippocampus is your major area of the brain. So here you can see there is, these are your control animals as well as this is disease and hyperdiet exercise as well as with metformin. And this bar graph you see here basically quantifies all the neurons. So this is the immunohistochemistry done by this new one is specific for mature neurons. So it will only stain mature neurons. And it is considered as a gold standard for looking at neuronal density. And you can see both in the pictures as well as when you do the statistics or count the neurons, there is a significant increase after the induction, and then there is improvement after exercise. And here you can see a more significance when you add metformin to the exercise groups. Same thing was again confirmed with Nissl staining. Again, Nissl is also is specific to neurons. So again, I will tell the students who are here, when you do a study, you should always confirm with multiple markers. Otherwise, sometimes what happens that you may get some false positive or false negative. So you always try to have multiple studies for the same parameter. So that is always good. So that will confirm your study whether it is actually happening. So we saw the same trend in both the cases. And then again, we looked at, since we have seen this increased cytokine levels, we looked at, so GFAP is basically a marker for astrogliosis. So it marks mostly the intensity of the color will determine, like if it is very high intensity, usually it is called that you have reactive astrocytes, which we call astrogliosis. If you have astrocytes but they are not reactive, in that case it will not stain that high GFAP. So GFAP is more specific for reactive astrocytes. And again, we try to quantify that. And again, this is for your CA1 region. Again, for information, your hippocampus has CA1, CA3 as well as the gyrus. The data I'm showing you is for the CA1 region. in CHE region also has similar data, but we are not having it here. And IBA1, again, this is a marker for microglias. So again, microglia, so all these cell types in the brain, astrocytes and the microglia, they have a good side, they have a ugly side. So normal microglia, it is, it is involved in phagocytosis. But reactive microglia, it will lead to inflammation, which the neurons cannot tolerate that much. So again, this IBA1, this is basically antibody that marks your activated microglial cells. So for microgliosis, and you can see here again in the CA1 region, there is microglial activation, whereas both your exercise as well as metformin is actually getting it not exactly to the normal levels, but very close to the control levels. So we did some more studies on looking at neuroinflammation, like this HMGB1. Again, this is marker for inflammation. Again, these are fluorescent immunohistochemistry studies. So we saw an increased level in the diseased groups as well as there's a decrease in exercise and metformin. CD200, again, it is a marker for basically vascular inflammation. There's vascular changes. CD200 is upregulated. So, like I again said, like when you have a hypothesis, you see various ways of how whether your hypothesis is correct or not. So, more markers you see, the better off you are. Next, we looked at okay, fine. There is a change in neuronal density. There is inflammation. There is some oxidative changes, reduction in acetylcholine or increase in acetylcholine stress levels. Now we want to say, okay, fine. There is protection after exercise, but how? How exercise is causing, leading to this protection. So what we initially our hypothesis is, or rather we are trying to test this is neurogenesis is one of the predominant ways by which exercise actually can, in a normal brain also, it can improve your neuronal health by causing neurogenesis. So we wanted to look at some of these pathways which are involved in neurogenesis, like WNT3 expression, beta catecholamine protein expression. And we saw this is basically a representative plot. And you use beta actin as a loading control, as most of tumors they are. So when you are doing a Western, you have to have a loading control. So we see a fold change in there's a reduction in WNT3 proteins. And again, these are hippocampal homogenetes. And then there's an increase after metformin exercise. So we don't see a very huge significance, but obviously there's a trend. For beta caroline, the same kind of trend we saw. Then again, we looked into some other markers. So I'll show you the signaling a little bit after this slide. So GSK3 beta, basically this phosphorylated GSK3 beta is the inactivated form, whereas this non-phosphorylated one, or rather, this is total GSK3 beta, and this is the phosphorylated. These are all the phosphorylated forms. So again, people have shown that irrespective of neurogenesis, GSK3 beta has a role with amyloid beta accumulation also. So phospho-GSK3 beta, actually dephosphorylation, like if there is activation of GSK3 beta, it may lead to accumulation or it prevents the degradation of beta. So here we saw there is a reduction in diseased animals, whereas again, though it is not as huge as in controls, there is an increased trend. And the same thing you can see in this quantification of the response. So here you can see this pathway here. This is one of the neurogenesis pathway that starts with the WNT signaling and it is inhibited by GS50 beta catenin, and DKK1 again, this is again an inhibitor of neurogenesis. So here you can see there is increase in DKK1 in your diseased animals, whereas it comes down after this single exercise as well as exercise plus metformin treatment. So it kind of suggests that somehow we are blocking the neurogenesis pathway in this diabetic animals, while in a control animal, as well as when you subject them to exercise as well as metformin treatment, we are reactivating these pathways to a certain extent. But again, their levels are not as like controlled. So we are not totally reversing it, but if a person with diabetes exercising regularly, probably he will have less chances of developing these conditions or this cognitive decline. So to summarize what I have shown you, basically again this is a work in progress in my laboratory. So prolonged diabetes can lead to neuroinflammation, oxidative damage, hippocampal lifestyle protein levels, reduced neurogenesis, and loss of hippocampal neurons, ultimately leading to this cognitive decline. Whereas exercise in combination with metformin seems to reduce the neuroinflammation as well as oxidative stress, which is again pretty obvious. Whereas another predominant mechanism by which it is protecting or is basically getting things back towards normal is by causing neurogenesis. And so these are the two intervention strategies we're proposing to basically not to totally eliminate neurodegeneration because of diabetes in this case, but at least to partially protect the neurons from the damage. So again, this is these are my students. We have most of this work that you saw here is done by Mr. Dikasundar. He's a PhD scholar, while the other master students as well as scholars here. And it's very hard work that I am showcasing. Thank you. Any questions, please?
Thank you, sir, for this demonstration of utilization of diabetes with dementia. And he has demonstrated quite elaborately that the relation of different pathways of occurrence of dementia and diabetes. So type 2 diabetes basically is a common form of diabetes that happens, and if it is unnoticed or uncontrolled, it may create severe problems like cardiomyopathy, neuropathy, and different other diseases, which is life-threatening. So I think, as I said it in my that welcome session of Banerjee, that Professor Banerjee, that it's much relevant topic with this conference topic, discussion topic. So he has demonstrated clearly. Now this session is open for question and answer session. Please, if any question is there, you can ask.
Anybody have any questions? Raise your hand.
Yes. Sir, is there any combination therapy of nootropics and antidiabetic agents parallel?
Yes, so currently anti-Alzheimer's drug you probably know is your rivastigmine, galantamine, as well as your amantine. So yeah, this NMDA receptor antagonist. So currently there is no established therapy as such. So you can put them on. Usually people are put on, and one more problem is diagnosis. A lot of times it gets undiagnosed. So like I said, after a certain age, people believe that, okay, I'm not remembering well, which is normal. But sometimes it may not be normal. And combination therapy can be given. Obviously, they will be on antidiabetics, but along with that, again, one of our study we are looking at mymentin. So again, this is part of the same MySEMA project where we are looking at the role of mymentin in combination with normal antidiabetics, whether that improves the condition or not. But currently there is no like first line agent kind of thing for people suffering from cognitive decline associated with diabetes. They are put on normal like whatever radiotherapy is there. No combination is given as such. So it's not well documented. Thank you.
Any others having any question, please? Anyone, please? Please.
Sir, your nice presentation. Thank you. I have just overview in general questions for the recent years. Type 2 diabetes, that is outcome of the metabolic disorders and dementia. So in our cycle, in the disease pathway, so is there any literature or any provision that which one is the upstream? It's type 2 diabetes will lead to the outcome of the it's leading to development of the dementia, or dementia to the patient who is suffering from dementia in connection to the different causes, maybe genetic, epigenetic, or something else of the food daily life, these are food habits. So it may prolong dementia, it may lead to the development of the diabetes type 2.
Usually, when you talk about depression, there is a clear literature on that. Some people say that diabetes is leading to depression. Some people are saying depression is that if you are depressed, then you will not maintain your lifestyle and you are more prone to develop this metabolic disorder. But when it comes to cognitive decline, it is usually, it starts from the metabolic problem and then it goes to the cognitive decline. Obviously, somebody who is at a very later stage of this, like you see that people who are suffering from Alzheimer's for a long time, obviously they cannot do anything on their own. They will need caregivers, they will need help to maintain their life. And that is why probably those patients are taken care of. So they don't develop diabetes as such. But obviously depression and metabolic disorder it can happen at a much younger age also. It is not subjected to age basically. So people who are having metabolic problems, they tend to develop depression. Again, people who are depressed, they tend to develop all these metabolic problems. So that is a more correlative or rather more complex.
So is there any literature in scientific that anti-dementia drugs may act on the treatment of the metabolic disorder type 2 diabetes?
No. Actually, like anti-diabetic, like I showed you the metformin.
Yes.
It has
have a role to
yes.
protect the
Yes. But whether this, because currently anti-dementia drugs, these are basically acetylcholinesterase inhibitors. Say they don't have much of a metabolic effect as such. Or you have NMD antagonist, which again does not have too much of metabolic effect. So we don't have that kind of drug that can modulate. We don't have it.
Another thing is that nowadays is a stress. It's a common factor in our daily life. And those who are anxiety, stress, sometimes it may lead to the development of diabetes, sugar, hypertension.
Like I said, this depression, anxiety, and metabolic disorders are much more correlated. Yes. Also, prolonged anxiety may actually cause some memory changes also. There is a literature on that. But that is a transient change. It should not develop into a dementia, but there is a temporary loss of memory or recollection in those cases. So there is a relation there.
Thank you.
Any question?
Yes, sir. How can we discriminate between absence of mind and cognitive decline?
Well, how do you define absence of mind requested?
So I have mistaken one thing in one place and I have finding them in other places. Just like that.
I don't know whether it's a scientific term or not. So there is a term called brain fog. So which is a temporary. So you are unable to recollect right away. But the fog, like a fog, it goes away. But dementia is a serious complication. Once somebody develops dementia, it's very hard to get it back or reverse it. So probably my answer to your question is this brain fog or what you're telling is reversible. But dementia is a serious complication. It's a life-threatening complication, and it's a lot of cases. It's like an end-life complication. So it's a very serious thing. So it cannot be reversed, and we don't have too many drugs. You can improve the condition. You can prolong it, but you just cannot reverse it. Right now, we don't have any drugs.
Any more questions from the participants? So I hope we do not have. So thank you so much, sir. We would love to felicitate you. We would love to appreciate your time, your knowledge, your expertise that you have shared. I will request our professor, Dr. Tapan Kumar sir, to felicitate him. Thank you so much, sir, for being here. Thank you for sharing your knowledge, your time. We would love to have you back with us. To be associated with you. Thank you so much. Thank you.
I also like to thank the JS Group for giving this opportunity, and I hope that since NIPER is coming to Pharmanecia very soon, so I hope we will have good collaborations.
We'll love to. We'll also support that.
Definitely, you're welcome to come to NIPER. Whenever we are organizing any conference, symposiums, or any short-term trainings, you're always welcome. Thank you.
Thank you so much, everyone. So, for the second day of poster session, we have completed our speakers' lectures. Now, we will head towards the eat break. But before that, we have our poster session. We know that we have two poster sessions. One is before lunch, one is after lunch. So, we will start with the before lunch poster session. For that, we are having our 4 judges for the poster session, 2 judges of each poster session. So we would love to felicitate, we would love to appreciate their presence of the judges in this poster session of this conference. For that, the judge we have, Professor Poloy Bhattacharya. He is the HOD, Head of the Department of Sister Nivedita University. I would love to call him in the stage. So please, if you come. And I would also request our HOD and Dean of Academics, Professor JISU-PHARMA sir, to felicitate him. We have our next judge, Dr. Nitananda Mondol. He is the professor of BCDA College. I would like to ask him to be on the stage, sir. And I would request Dr. Payal Chakraborty, convener of this conference, to felicitate him. Thank you so much, sir. Thank you so much, ma'am. We have our next judge, Dr. Shashubra Ghosh. I would like to ask him to be on the stage, sir. And I'll request Professor Komalakanta Roy, sir, professor of our department, to felicitate him, sir, please. Thank you, sir. Thank you so much. And our fourth judge, we cannot forget, who has already with us yesterday, Dr. Parthu Palit, Assistant Professor, Aftam University, Stilsor. Thank you so much, sir, for judging our poster yesterday, and we would love your assistance today as well. Thank you, sir. So now I'll request all the students, all the participants to have the tea break, and the participants who have the poster session up to the serial number 53. They can, after having the tea, they can go to their designated place in the third floor. That is the floor like 400 floor. So you can go there and you can be with your poster, because judgment will be starting just after 15 minutes of the tea break. So the tea break will be for 15 minutes. After that, the poster session will be started. So I'm requesting all of you to be there on time. Thank you, everyone. We will meet again after the lunch. Thank you. Okay, for the lunch, there's an announcement. Everyone, please. All of you, please have attention at the back. After the poster session, 2 poster sessions. So we'll be having our poster session now after just 15 minutes of tea break, and we'll be having our lunch time. So for the lunch, you have to go to the 516 room number, 516 room number, that is in the 4th floor. After your lunch, you will get 30 minutes of time to complete your lunch. After your lunch, there will be again a poster session. Once your poster session is done, there will be a cultural session and a valedictory program here in this auditorium. You all have to come to the valedictory session to know, to see the cultural program, obviously, and to know who has come first, second, who has qualified in this poster session for the result. So after the second poster session, please do come back to the auditorium for the final session of our conference. Thank you, everyone. Thank you so much. So the valedictory session will start around 4:30. 4:30 is the time you have to come back after the second poster session. Thank you.
হ্যাঁ তাহলে তুই সাথে থাকলে আমারটা একটু যেতে ভাব কি হবে বাড়ি